So good afternoon, everyone. Second attempt. Welcome to the people that are attending in person and the people attending online. This afternoon, and thank you very much for your patience with the load shedding, but we thought we just wait that the generator kicks in before starting. So this afternoon, we are lucky to have Dr. Fidele Tugizimana with us. Fidele, sorry, I hope I pronounced it more or less right. If not, you are welcome to mispronounce my surname anytime. Fidele studied biochemistry, first in Rome, and then I think at the University of Johannesburg, where he completed, I think, his PhD. And now he's the deputy head of the biochemistry department of the University of Johannesburg. And at the same time, he also works in industry at this company called Omnia. Yeah. Fidele is the president of the Metabolomics Society of South Africa, and he acts on the board of the International Metabolomics Society as well. And that brings you straight to the topic of the talk, because he's going to explain to us what this has to do with big data and computational strategies. So Fidele, thank you very much for being with us this afternoon, and welcome, and we're looking forward to your talk. Please.
Thank you so much for kind introduction. I'm really glad to share with you some points, some ideas, some studies on metabolomics. When I was invited to give this presentation, I was told the community is mathematicians, computer scientists. So I thought to share with you the field of metabolomics with highlighting a few aspects or elements where mathematicians in terms of chemometrics or big data analytics and computational scientists, or where they can really overlap in the terms of metabolomic studies. So I hope at the end of this presentation, I would have really got your attention in terms of extending collaborations, especially with your expertise in terms of in the mathematics expertise and also computer science expertise. So in this presentation, I will just do these three aspects to try to define or explain what is metabolomics, and also the community of metabolomic scientists internationally and here in South Africa. And also I will spend much time again on sharing with you studies, metabolomic studies that we have done, in which data analytics is used and computational strategies. Can you hear my voice well? Or just double checking. Yes, I assumed yes. Okay, fantastic. So what is metabolomics? If you look here, those who have assumed, all of us, we have a bit of biological knowledge in basic biology, where we understood the biological system and the way it functions. One of the things that define functionally the biological system is the metabolism. And metabolism, understanding the classical way, is driven by the information that are captured in genes, encoded in genes, which are then passed on or transcribed in other genetic material, and then translated in proteins, which also have different functions, and leading often to the synthesis of what we call small molecules. So these small molecules are known as metabolites. So we can say we have a gene space where we have these different genetic layers that leads to a chemical space where we have these small molecules that are more closely or that describe, if one may say, the phenotypic state of a biological system. So this small molecule chemical space can be endogenous from the organism itself, or they can be from outside. For instance, when I drink a cup of coffee, I drink a number of small molecules that comes to my system and they get processed. Or when you go to the doctor, you get a drug, that's a small molecule that gets into your system and get processed and leads to a certain effect. So metabolomics or metabolome is this entire collection of this small molecule, what we call different terms that scientists aim to coin, molecules, or a space of different small molecules. And the study of this chemical space is what is known as metabolomics. And to make things more complex, if you were to understand the metabolism, it's really a multi-layered complex system where you have different genes that can lead to different proteins that can lead to or have effect or define the way this chemical reaction will take place. But also at the same time, this chemical reaction does affect the expression of these other levels, be on a proteome level or genetic level. So the metabolism of a living system define what indeed we see as sitting here, listening to this presentation, interacting with you. There's all kinds of chemical reactions that are taking place. So to zoom into this area of the metabolome, we gain quite a lot of insightful information that tells us not only the effect of the outside, but also the imprint, genetic imprint of the organism understanding. So in a very simplified way, we can then understand metabolomics as that the science that study the metabolism with a focus on the small molecules known as metabolites. So the term metabolomics was coined in 1998, 1999, and there are different Robert Hall, Oliver Fien, Jeremy Nicholson. These are some of the key fathers of the term metabolomics or metabonomics. The science have grown quite a lot. This is what I just did in last year. If one were to look just 20 years back with publication that comprise the term metabolomics, it's really an exponential growth. The science itself has grown so fast, and it has really shown a certain impact in terms of the fundamental research, but also application. However, despite the advancement of metabolomics or the science, we still really not have a comprehensive understanding of that chemical space that I've shown. For instance, in a plant, we estimate there is more than 200,000 metabolites, but we really know very little, a very small fraction. In a human, estimation of more than 100,000, but we really know very little of that. So it is still, there are still quite a lot of bottlenecks, challenges, and efforts in terms of advancement of science. How do you conduct a metabolomic study? There is a certain workflow that have been proposed. There is different methodological ways to carry in a standardized manner metabolomics. What I wanted to, for the sake of this presentation, to bring to your attention is this element of multidisciplinarity of the metabolomics. It's really involves different disciplines: biology, chemistry, statistics, or data analytics, and computer science. And it's really not one man, one person job. It's really a team effort. I doubt anyone can be really an expert in these all disciplines, but we really, if you have a good team, with different expertise in this different discipline, the success of your study is really guaranteed. So the workflow is a multi-step workflow involving a sample preparation, acquisition of the data or sample analysis, and then much of the time is also spent on data mining or interpretation to gain a sense of biological insight. Put in the context of big data analytics and computational strategies, in these different steps of metabolomics workflow, there are different technologies that are being applied in sampling, sample preparation, with now 4IR tending to use 4IR technologies to minimize human errors in sampling, data acquisition. There is advancement in technologies where we see analytical techniques such as mass spectrometry-based or NMR-based that are equipped with a kind of analytical intelligence that helps to automate or acquire quality data. And data analysis, where we spend quite a lot of time, where we see then the use of big data methods, machine learning, deep learning, and also computational strategies that helps to mine and interpret the data that one has acquired. I have this is a diagram that points out to some of the computational tools that have been developed that are being also used in metabolomic studies. I will come to some of these in this presentation. If we were to look then on the data side, big data, metabolomics studies generate quite a big data in terms of volume, in terms of velocity, and the main analytical platforms that are used are mass spectrometry and NMR. These generate really quite spectral data, big data, that we can really name big data in terms of the volume and the size and complexity. And to then make sense or extract information from this big data, we do apply different algorithms. So here comes in the mathematicians, expertise of mathematicians, different algorithms to process, to clean the data, and also some of the econometrics methods that are classically used. There is unsupervised and supervised analysis, PCA or OPLSDA. And then we want to make sense of the features that we have generated, or the model that we have generated, to understand in terms of biology. There is also growing, as we generate quite a number of huge data sets. How do we handle this data set? How do we deposit this data set? And also, how can we reuse this data set? There is a cloud metabolomics, a team effort that is being put towards cloud metabolomics, where we hope to generate, to build a cloud system where different tools can be accessed, and also where we can deposit data sets that are generated, and maybe have a chance to reuse those data sets to gain more understanding in terms of the question at hand. Computational metabolomics, I would recommend for those who are interested to see more in terms of the computational strategies that are employed in metabolomic studies. There's a recent review paper written by these two great people, Tim and Justin. It really highlights the current state of these computational metabolomics tools that are being developed. Where are they used, the limitation, the advantages, etc. So there is, if you want to look at the publication 20 years back of the computational metabolomics term, it's really, in the last 2 years, it's really in a huge exponential growth. In our research, we do employ a lot of computational metabolomics tools. They provide the way to gain insight or extract information from spectral data. This omics science has then gained a huge momentum, and it has application in different fields, nutritional, metabolomics, disease prediction, medicine, drug discovery, biomarker discovery, personalized medicine. These are some of the areas in which metabolomics is being applied. In our research, we focus mostly on plant metabolomics, with a focus on drug discovery, natural product discovery, and also on biostimulant, plant biochemistry in context of agriculture. I have put here some different centers, metabolomic centers across the globe. There's one Northwest with a focus on human metabolomics and human aspect. At UJ, we focus mostly on plant metabolomics. And in other areas of the globe, I think this is UK, where there's a Phenomic Center, Australia, etc., etc. So metabolomics has grown in the last 20 years so rapidly, and it has found impactful application in different fields, not only just research and academic environment, but also in industry or other areas. The community of scientists across the globe have come together to form a Metabolomics Society, which really the mandate to promote the growth and development of the field internationally. And one of the major events that we organize annually, it's a Metabolomics conference. For this year, it will take place in Canada in this date, June 18, 22. And that a platform where there is a showcase of different advancement, different recent development in the science. And also it gives the scientists across the globe to come together, to network, and keep advancing the science. Here in South Africa, we have the association, Metabolomics South Africa, which is an affiliate to this International Metabolomics Society. So far, we have more than 300 sign-up members, and it was established not long ago, in 2018. And these are some of the institutions where the metabolomics research, these studies are being conducted, also in different fields, human and plant, and also infectious disease, Victoria, TUT, where they do quite a lot of natural product research, and Venda also. I didn't put the picture here, but there's different institutions where the metabolomics is being applied here in South Africa. And we have online visibility with a website, which will be, today I think it's down, but maybe tomorrow it will be up running again. And also on YouTube, where we share also different workshop or presentation or on different topics in metabolomics from different institutions, and also on Twitter. So you can really, if interested, you can link with us and follow us what we do across the country. For my research then, what I do mostly, when I say I do, it's actually my team, the ones that do quite a lot of the work. I focus on different areas, the methodology involved in metabolomics, with a focus on data analysis or data acquisition, and also exploring computational tools that are being developed, and with the hope to participate in developing these computational tools. And I guess here, the community here, the mathematician, the computer scientist, it's really an invitation for collaboration. I do also, with different colleagues and collaborators, look at natural product research with intention to understand South African medicinal plants, which are really rich, and less explored and have very potential for health benefits. I then work with Omnia, as you saw, as Paul said, where we look at what the biostimulants, microbial, non-microbial, with a contribution towards how this formulation can be made, and also the program of application of this formulation. So I will give some examples in these different areas, also highlighting the use of those computational tools, data analytics, and the information that we gain from such type of studies and how they can be used. So for methodology, as you see in different slides, there is a term molecular networking. It's different methods that have been developed to really find a way that we can extract information from spectral data that we generate. So this is based on a simple philosophy that those small molecules that I showed you, the metabolites, which are really chemical compounds that are found in our system endogenously, or what we take in the chemistry. If the structure is similar, if you fragment them, meaning you chop them into small pieces, you break them into small pieces, if they are structurally similar, they will generate a similar fragment or related fragments. So based on that principle, or if one can think in a simple analogy, if you have cups, different teacups, you break them, and bottles, you break them, the cups, because of the way they are structurally similar, they will generate similar fragment. The bottles of Fanta, for instance, or coffee, they will generate similar fragment. So if one can take those fragments and put them together, can reconstruct the structure. We can reconstruct, this was a cup, this was a bottle. So similar reasoning on the small molecules. If we use mass spectrometry, it's a tool, analytical tool that we use to analyze these samples, and you fragment those molecules, you generate different fragments, and we can then pull together those fragments which are represented on the spectrum, and the ones that are put similar can be put together, and you can start rebuilding the connection based to structure similarities. If you can then put together create different clusters or different cluster nodes due to structure similarities, that gives you an advantage to be able to annotate. If I can know this is a cup, I can know that in that cluster, everything that I have there, it's cup related. So if I can know one structure, be able to name one structure, I will be able to put a name on or other structure in that cluster. So that's the simple reasoning and philosophy behind molecular networking methods. So there are many, of course, that's very simplified. In reality, things are more complex than that. Computational scientists are really crucial in this aspect because they are able to tell this is computationally done. They are able to write these computational languages that can help us to really put this information together. So the other tools that are being developed, computational tools, it's what we call MAST. So this is a tool that helps to really search the database. So as we generate different spectra, different structural information, and we deposit them in the database, one would wish to have a tool that can easily search where are those molecules and how are they connected to other molecules. So there are different MAST tools that are being developed, food MAST, with a focus on the food molecular composition from food material, microbe MAST, which you have been involved in, that with a focus on metabolites or molecular composition from the microbes. The other methodology focus that we do is machine learning methods that are being reintroduced in metabolomic studies. As I said before, we do generate quite huge data sets, so we need really to use tools that can help us to maximize the information that we extract from those data set. I will just give an example here. It's a dissertation from this Fidele Tugizimana. She's currently doing her PhD. She completed her master's. What we did in this preliminary study, if I may call it, we assessed different machine learning methods in mining plant metabolomics data. So we looked at 6 machine learning methods, which are listed here, and we assessed them in terms of classification accuracy and variable importance. And we looked at linear, non-linear methods, and though we used this not really huge big data set, but the data set we used could really give us an impression that non-linear machine learning method performed better than linear, the normal PLSDA that we normally used with PLSDA, but there's also the other version of orthogonal PLSDA, which is also used for classification analysis and variable extraction. So future studies following this idea that we have is to perform this machine learning method across different main data sets, and also to assess the influence of data preprocessing on the performance of this machine learning method. And also, this was a binary study, so we would be happy to look at multi-class context, how this machine learning method would perform. As I said, why one would want to do such type of exercise is to really be able to understand, to gain a model or a method that can handle big data set, and also that can extract, maximize the value of the information that we gain from untargeted metabolomics data sets. So this is an ongoing study or idea, but now it's on pause, but it will carry on. So for natural product research, what we do, we get medicinal plant material, we apply, extract those small molecule in a more standardized way, and we analyze those small molecules, mostly on mass spectrometry. We use mostly mass spectrometry, but one also can use NMR. And the spectral data that we have generated, we apply a number of computational tools, a machine learning method to extract information to be able to understand the metabolome of this medicinal plant. As just a quick recap, as you may know, the traditional way for drug discovery or natural product studies, it's a kind of fractionation way where you extract plant material or any source, be a marine material, you extract the metabolite or product or compounds from it, you get a crude extract, and then you test the activity of the crude extract or different fractions. And that would lead you to a fraction that is more active, that has high bioactivities, and you would assume that's where you have your natural product of interest, and you can perform other structural elucidation. So that's it, time consuming, and also the disadvantage is that you may end up re-isolating or extracting the same type of compounds, and you miss out quite a lot. So applying metabolomics gives us an opportunity to be able to map this chemistry of the natural product to get a more comprehensive map or landscape, which we may call a metabolome. And we can have, if you have already a map of a city, for instance, you are able to say, okay, I'm going to Santon, I'm going here, I'm going there, or you can be able to tell where are different functionalities or potential activities then we do apply also. You can add other computational way to predict activities and then biological assays, etc. So some of the computational tools, emerging ones that are actually being developed that we use often, I have listed them here. As you will see, it's mostly those molecular networking tools and other in silico tools that helps to enhance the identification of those small molecules. So one of the study I would love to share with you very briefly, it's where we looked at the Momodica plant, which from Venda, a study done by Anza. She's completing her PhD. And what we found, we applying these computational tools, remember, as I said, collecting structural information, you can be able to put together structurally similar compounds. And here you can see different clusters of structurally related compounds. So this is really computationally done. And we were able to map, to annotate the metabolome of Momodica plant to see different kind of classes, flavonoids, glucosides, amino acids, terpenoids, etc. The other two studies in natural product context research, it's cannabis and also helichrysum done by Akona. She completed in her, she did this in her master's studies and she has started her PhD. And the other one of helichrysum, it's Matsewa Lepatsi, who is completing her PhD with the same idea, the same approach. We want to know the metabolome of these plants, and then we test the here in this study. What I wanted to point out in terms of computational scientists is the use of molecular docking, which is really a computational prediction of the activities of different compounds that one would have extracted. So as we saw here in the Akona study, we cannabis, we did also add on, in addition to computational prediction, we did some cancer assays where we really found that the extract from cannabis does induce apoptosis in cancer cells. So here also, in Matsewa, it's a more focus on cancer also. If we predict, here it's an example of some of kinases that are involved in cell proliferation. Some of these compounds then inhibits the activity of this protein. If this protein can, by binding, change the conformation, one would assume the activity being lost. So if the activity lost, so this would then lead to death cell and to the apoptosis, which is the cell death of the cancer cells. So this just to show you the use of computational methods which are familiar to you in metabolomics research, how this also can lead us to more insightful and impactful results. So the other thing I said I do, it's working in the Omnia, where we look at the biostimulants, biostimulant being microorganisms or organic matters that are found naturally in soil, using them to the benefits of crop plants for in promoting growth or enhancing the growth of the plants, or also improving the defense mechanisms of the plant. So the question is, how are these biostimulant function? What are the mechanisms? What are the mode of actions? So our research working with Omnia is to really elucidate those mode of actions or mechanisms, and also how the plant responds to these biostimulants. And for the Omnia, such information helps them for formulating this biostimulant, but also coming up with a more precise application programs. So an example here would be this microbial biostimulant based on Bacillus strains, where we look at the chemistry of these microorganisms with the reasoning that if the microbe interact with the plant at a root level, it will interact or communicate in chemical language. So what are the chemistry that these microorganisms have that are involved in a chemical communication with the plant, which would lead them to either enhance the defense, or if what we can call vaccination of the plant, or priming of the plant, or increase enhanced growth. So we do the same. Here we just did a liquid culture of this bacterium and looked at intracellular metabolome and extracellular metabolite of this bacteria. So we extract the metabolites from these cells, analyze them on LCMS system, then apply computational tools to annotate the metabolome of those microorganisms. And we were able to see different chemical families of these that define the metabolic landscape of all these microorganisms. And we just to draw attention here on lipopeptides, which are really antibacterial compounds that are secreted by this bacteria, and they have different implication or role to sort of defend the plant, but also to prime the plant for defenses. So this study has been done by Lerato, who is completing also her PhD. So as I said, why would want to do such study? If you were to look at this diagram here, it really gives an idea how complex that chemical communication is between microorganisms and the plant root. So if one can understand these chemistries of these microorganisms, you would be able to design a more precisely the formulation that would lead to the effect that you want to see. The benefits are, some of them include defense priming, as I said, nutritional, via nitrogen fixation, etc. So these are some of the team members that are involved in microbial metabolomics. Lerato PhD, Teboho also doing his PhD. Major completed his master's, and Motumiseng also just completed her master's. Then that's on the biostimulant side, but then on the plant side, the question is that what are the responses at cellular level and molecular level of the plant to this treatment? Why one would want to understand those plant responses to biostimulant treatment? It gives us an idea, as I said from the start, that our life, if one was to reduce it to a small molecular definition, is really the metabolism, how our metabolism happen, or what's going on at the metabolism level. And that really gives us an idea whether it's a positive, there's a benefits, or there's a negative effect. So if we can understand how the plant at the molecular level, at the metabolism of the plant, how the plant is responding to the biostimulant treatment, we can extrapolate the effect or the mechanisms of the biostimulant. So I will give example of microbial and the non-microbial. On the microbial, there are these three, it's one big study that was divided in small studies. Done by these three students, Lerato in her masters, Khalaret also in her masters, and Motser also in her masters. So we looked at a microbial biostimulant, which was a mixture, a consortium of different bacillus strains, and then which were then in a greenhouse setup, controlled environment, where then we applied that consortium on maize plant, and we subjected the maize plant to drought conditions, stress condition. Then we investigated the response at the metabolism level of the plant to be able to deduce what are the mechanisms of this microbial biostimulant. So these are the publications that you can see we resulted from this study. So on the metabolome level, we applying again those computational tools. We really not just to for the sake of time. Applying those computational tools that I've shown you, that really helps us in annotating these small molecules. We were able to annotate the metabolome of the maize plant, comprising different classes of metabolites. And we can also, there's a bioinformatics tools that we can then investigate what are the pathways. The metabolism described in terms of biological pathways. So here we can look we were able to see enhanced defenses in different pathways that be impacted, also growth promotion in different pathways that be impacted by the application of the biostimulant. So this led to coming up with a model that could schematically or could postulate or postulation of different mechanisms that this PHPR, plant growth promoting bacteria, induces in a maize plant. So we did also in Motsewale Patsi study, complemented the metabolomics work with genetic studies, gene expression and the methylation, which complements or correlated well with the changes that we saw in the metabolite levels. So it's a different changes that we could see in the sap, which is really transportation components in the plant, and the leaves also, the changes in the different pathways, which have these physiological events or drives these physiological events that are benefits for the growth of the plant and also for the enhanced defenses in the plant system. For non-microbial, those were microbial, which are living organisms. For non-microbial, these are, for instance, silicon-based or organic matter like humic acid. We did also some studies. The study was done by Fidele Tugizimana in her master's, where we looked at the silicon-based and looked at the tomato plant under salt stress to then see what happened at the metabolism level. So to cut the story short, we could see that different pathways were impacted by the application of that silicon-based formulation by stimulant, which include phosphate, pentose pathway, TCA, and other fatty acid biosynthesis pathways. So putting into correlation to see this correlation network, to see, because these pathways can be impacted, but these pathways comprise different metabolites that are correlated in different ways. So one would want to see in terms of the regulation of these changes at the metabolism level, how are these metabolites interconnected independently of the biological pathways. So correlation network, which is also really a computational algorithms that helps you to do these calculations, involve mathematics also involved. We can be able to tell the change in this metabolite, increase, decrease, or no change. How are they interconnected, independent of the biological pathways? In this study, also, we came up with this postulation that described the mode of action of the silicon-based formulation, defining how the plant responds, which indicates the different activities at the molecular level that silicon-based biostimulant induce or activate in the plant system. So we do these studies in a controlled environment where we then look at different concentration application, areas of application, timing also. All of that are taken into consideration, and it gives an idea, and one can go back to be able to design well the formulation or the application program. In the context of our daily life, where the world wants to be a better place, all these studies that we do here on biostimulant are very significant in a way that we want really to, with the climate change or increasing population globally, the crop production system is really under constraints. So there is much demand in terms of food production, but the production then on the side of the agriculture sector, it's really under stress, if one may say. So having this biostimulant put into place, coming to market, it really has a great impact on agriculture sector as it promotes growth of the crop plants that we rely on for survival, for our daily life, and also it minimizes different stresses. It makes the plant be able to respond effectively to different stresses that arise in the environment, be abiotic stresses or other biotic stresses. And this really align well with the sustainable development goals, such as zero hunger, reduction of poverty, etc. So this is a newsletter published in UK, where also we highlighted in collaboration with the people from UK the benefit of applying metabolomics to really how this approach is impacting the biostimulant industry. I hope in this really very long presentation, I did convince you that mathematicians in the line of big data analytics, computational scientists are really people that we would love to partner with in this metabolomics journey, because we rely much on your expertise in terms of machine learning, deep learning methods, artificial intelligence, computational languages or computational frameworks that can really help in extracting information from the data that we generate, and down the line also be able to gain biological insight that we can, in a different context, in different environment, be able to translate in a key effect that has a benefit to our human lives. I would like to end with really gratitude to this team of mine, because as I said, I tried to indicate on all different studies, different people who were key conducting of these different studies, the PhD candidates, 8 of them, 9 of them, and also master's students who are working on different projects. I would like to end with a great thank you to the collaborators nationally and internationally that really tap into the expertise, we collaborate on different projects, and we learn a lot from them and from each other. Thank you so much. We'll be happy to answer.
Thank you very much, Fidele. Thank you very much for the very nice, informative overview.